Welcome, everybody. Thank you so much for joining us tonight for another Sharp Minds lecture. Sharp Minds is a lecture series by the Fleet Science Center here in San Diego in beautiful Balboa Park that we normally host on the first Monday of each month. It would normally take place in our beautiful Dome Theater at the museum. Unfortunately, due to COVID, the museum is closed at this time and will remain closed, it looks like, until the end of the year. So we will continue with these talks online. And we're really happy that we have that opportunity to still bring you these exciting science talks, even though unfortunately we cannot see each other in person. So I hope you're already connected to us through our newsletter so that you get the notifications about the upcoming talks, but they're also on our website, rhfleet.org. So if you remember, first Monday of every month, that's the time for the Sharp Minds lecture series. But we have many other science talks, as you might have seen in the presentation. We do conversations with scientists, which is our Sets in Science program. So that will not be a traditional lecture. We try to have a verbal explanation by the scientists followed up with a more extended Q&A or conversation. And then we also have our Sky Tonight show, which is our planetarium show that happens on the first Wednesday of each month. So we hope we have many topics and subjects to engage you in with science. And I think that concludes my introduction. There's one more thing I'd like to say. So we have enabled the chat for tonight, which we normally don't do, just, for Zoom security purposes and stuff. But our speaker tonight is excited to engage you. So she will have some questions for you. So you can use the chat function to communicate with her and me throughout. We still have the Q&A function for the questions. It's a little bit easier for us if you have questions after the presentation, if you could use the Q&A function. This way all the questions are in one spot and I don't have to look all over for the questions. But either way, I will make sure that I keep my eyes out for both. And then I will also put a little link into the chat for all of us, which is a link to a survey which I would really appreciate you guys filling out at the end of the night. You can click on it already. This way it opens up the Google form, the survey for you already in a different window so you don't forget. Because this one is a special survey. It's part of the If Then initiative, which is an initiative that highlights and supports women in science, and Samantha is part of the If Then initiative. She is an official If Then ambassador. There are If Then ambassadors all over the United States. So they send us a little link to a survey that they would like us to use for tonight's talk, because Samantha is an If Then ambassador. So thank you so much for taking that survey at the end of the night. I'll try to remind you again to do that. But with that, and without further ado, I'm going to turn it over to Sam to lead us through some great information about the Cabrillo National Monument in our backyard.
Yeah, exactly. One of the most beautiful places in San Diego. I might be a little biased, but I think many of you will agree. Hold on one moment. I'm going to share my screen and we'll get started here. So let me get to my presentation.
It really is a beautiful spot.
For sure. All right, can everyone see that? I hope you can. Yep. Hi, everyone. I am Sam Wynns. I am a conservation biologist and science educator with the Great Basin Institute and National Park Service here in San Diego at the beautiful Cabrillo National Monument. And as we mentioned earlier, I'm going to ask you some questions tonight. I find it better for participation, right? It keeps you awake and alert and keeps your creative juices flowing, your thinking caps on. And also, science is a collaborative effort, right? So we're going to collaborate here together tonight. So my first question for you, we're going to test it out, is what do national parks mean to you? I'm sure most, if not all of you, are familiar with our national parks. And let me get this chat function open here. So what do national parks mean to you? So you can go ahead and type in your answer in the chat box. We'll give you a second here to type that in.
Can I participate too?
Yeah, of course. Please do. It's open to all. I feel like we need some Jeopardy music right now.
I know, right?
Do, 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 do. Yeah. That's all I will sing, I promise. It's an earworm, so it won't get stuck in your head forever. Yeah, I'm seeing beauty, relaxation. I would agree with that. Anyone else want to chime in here? Don't be shy. Great beauty. Love it. I'm going to give it just one more moment, see if we get a couple more answers. All right, we're going to go ahead and move on. Yeah, beauty and relaxation, you're right. These words characterize our national parks. In fact, our national parks can be defined as the locations of our nation's most important cultural, historical, and natural resources. And the National Park Service, the organization that I work for, is tasked with preserving and protecting these sacred places. Oh, there we go. But that is easier said than done, as you can probably discern from this map, right? There are approximately 420 national parks in the national park system. And that includes national parks, national monuments, national seashores, national battlefields, etc. And they're scattered all across the United States and also in US territories like Puerto Rico and American Samoa, right? And all told, that's about 85 million acres placed in the National Park Service's care. So that's a pretty mighty task to preserve and protect that. On top of that, every national park is different from the last, right? Different topography, different climate, different resources, different neighboring communities. And just to name a few of these differences, right? So each park has to be managed in its own unique way to reflect its own unique characteristics. So when you think of all of that and put it all together, those 85 million acres, all the different types of national parks out there, it is a super mighty task to do it. So how does the National Park Service preserve and protect these incredible, important places? Well, in a variety of ways. The National Park Service is organized into different departments that focus on one piece of the conservation puzzle for their specific park, right? So here we have an example of the organization chart for Cabrillo National Monument, right? So you can see at the top, we've got the superintendent, Andrea Compton. Now she's kind of the head honcho, right? She's in charge of all of the departments put together, makes the big picture decisions. And then underneath her, we have the chiefs of the different divisions, right? So the division administration, well, how do they preserve and protect? Well, by keeping things running, right? By cutting the paychecks for everyone else who works there, right? That's how they help to preserve and protect our national parks, right? Division of facilities management. These keep things flowing at the park, right? This division keeps things flowing at the park. So cutting new trails, maintaining trails, putting up guide fences, these sorts of things that help protect our resources, right? Prevents people from trampling or going off trail. That's something that the facilities management does. We're going to skip over interpretation education really quickly and go to Division of Resource and Visitor Protection. These are the law enforcement rangers, the rangers that carry guns, and they do things like preserve and protect by making sure people aren't poaching, right? By making sure people aren't spraying graffiti on our beautiful resources, right? And then last we get to the work that I do for Cabrillo National Monument. And as a conservation biologist, it's my task to preserve and protect the native species of plants and animals in the park, to safeguard those natural resources and the ecosystems therein. And that is what the Division of Natural and Cultural Resource Management and Science does. It employs scientists like myself, like biologists, environmental scientists, or even archaeologists, because we can't preserve and protect what we don't understand, right? So we need to go out and study these resources to be better land managers, to be better conservationists as a whole. So here's some pictures of me doing just that. How we study is through something called inventory and monitoring programs, where we catalog biodiversity and take data over time to compare and contrast over the years. Our national parks are fairly unique in that they are mostly intact wild lands, right? So you can kind of think of them as a living laboratory. And at Cabrillo National Monument, we currently study most groups of native species, okay? So birds of all kinds, so anywhere from our seabirds, shorebirds, to songbirds, and even our amazing peregrine falcon pair. We study our reptiles and amphibians. We study plants, we study bats, and all of the marine organisms in our rocky intertidal ecosystem. And of course, each group must be studied in a way that reflects its lifestyle and life cycle. It wouldn't make sense to devise an inventory and monitoring program for an octopus the same way you would study a bird, right? I mean, one is a marine invertebrate, one is a vertebrate that flies, right? So here I am monitoring the different groups at the park. So you can see right here on the right, in the time of COVID, because I'm wearing a mask, right? I am monitoring our terrestrial or land birds. I've got my binoculars and my field gear to do that. In the middle and the lower left-hand corner, I am monitoring reptiles and amphibians. And you can see I've got a little lizard in my hand. That's a Western or Great Basin fence lizard, the most common lizards you're gonna find around San Diego. And no, it is not dead. I actually fell asleep in my hand while I was taking some data on it. So that's why I have such a big grin on my face. Then in the lower left-hand corner, I'm helping another wildlife biologist as we're about to take some data on a little young snake there. And then in the upper left-hand corner, here I am in our rocky intertidal zone. I'm measuring an organism that I'm going to talk about in a little bit called an owl limpet here. And in fact, funny that I touched on that, I'm going to focus on two groups tonight because I could talk forever about how we study all these different groups. So I'm just going to kind of hone in on a couple of these groups, and that is how we study our rocky intertidal organisms, so the upper left corner, and how we study our herpetofauna. Fun fact alert. Herpes is the Greek word for a creeping thing. So whenever you hear herpetology or herpetofauna or herpetologist, right, a herpetologist is someone that studies creeping things, also known as reptiles and amphibians. Okay, so I bet you didn't know that. You can go around, that's your new party trick, and tell someone that herpes means a creeping thing. Okay, so how do we monitor our reptiles and amphibians, aka creeping things? Is by using a tool called the pitfall array, which is depicted here. And these arrays are basically buckets that are buried in the ground up to the lip. And the configuration of the arrays is that there is one center bucket, which you can see in this rightmost photo here, with 3 arms of fencing that lead away from that bucket. So you can see there's arm number 1, arm number 2, and arm number 3 coming off of that, right? And at the middle of each of those arms is another bucket. And at the end of each of those arms is, guess what? Another bucket, for a total of 7 buckets, 1 in the center and then 2 on each arm. Are you with me so far? Now, most of these buckets are closed, but for 1 week out of every month, we open them up and then we set the lids like so. And I'm going to demonstrate here with a little bucket. Andrea, let me see a thumbs up. Can you see this? Can you see the bucket? Okay, great. You can see there's little legs on the top of that bucket. We open up the bucket. We set the legs like so, which creates a gap for little creatures to fall into. But these buckets wouldn't be very effective, right? Creatures would not fall into them if they were just kind of buried willy-nilly about the park. So in comes the guide fence that I mentioned earlier, right? You can see there's fencing between the buckets. And what it does is it guides creatures into those open buckets. So I'm going to demonstrate here on my hand. And let me just see if I can change it so I can see myself there. That makes it easier. Okay, so pretend my hand is the guide fence. And this little plastic lizard, no real lizards today, unfortunately, comes along, runs into the fence. My next question to you, which please type your answer into the chat. which direction do you think this lizard is going to go when it runs into the fence? What do you think? Left, right, up, down, where is it going? And while you're typing in your answer, I'm going to take a sip of water. Excellent. I'm saying left or right, and you're absolutely correct, right? It's going to take the path of least resistance, right? So having to climb over the fence, having to dig under the fence is going to take more energy. So 9 times out of 10, that lizard will come along, bump into the fence, and just run along the fence, and guess what? Boop, right into the bucket. Okay, now it doesn't matter which direction it turns because there'll be a bucket on either side. That's why the pitfall arrays are configured the way that they are. Now, my next question for you is this. Are lizards and snakes the only things we can catch in the buckets? And do we only catch one of them at a time? So what do you guys think? Are they the only things we can catch in the bucket? Do we only catch one at a time? Giving you some softball questions here tonight. It'll get harder as we go. What do you think? Aha, I'm saying rodents. You're absolutely right, right? Basically anything small and terrestrial, we can catch in our buckets, right? So we do catch rodents. And I'm gonna bring my demonstration bucket back here. We have some things inside these buckets to be better stewards, right? Because again, we're capturing these animals to take some data on them, but we don't really want to injure them, right? We want them to survive. We're there to preserve and protect them. So for instance, we have this very fancy solo cup with cotton batting in it. And that is actually not for reptiles and amphibians, it's for rodents, right? They like to stay warm at night, so we give them almost like a little comforter. And I always know I've caught somebody because I pack in the cotton batting like this. And when I come back in the morning, if it's partially out, I know there's somebody sleeping inside or hiding inside, right? Sometimes we catch more than one animal, unsurprisingly, I'm sure. So what we do is we have these little hides in there. We have these PVC tubes. This gives them a place to hide from one another because guess what? Occasionally we catch predators and prey together inside the bucket. Then we also carry a wet sponge in the bucket because we do have one amphibian species at the park, the garden slender salamander. And of course, like almost all amphibians, it needs to stay wet, right? It breathes primarily through its skin. So we want to give it a nice wet sponge that can survive overnight. All right, so once we have caught our species in the bucket, we're going to take some data, as I mentioned earlier. We're going to take data like weight, length, we'll sex it, we'll see if it's an adult, and condition, those sorts of things, right? But at this point in time, you might be wondering, what happens if you catch a really large snake, right? This is only a 5-gallon bucket. Won't the snake just come right out? And you know what? It will, in fact, just come right out. So we have something else called a snake trap. And it's this very fancy tube of chicken wire with 2 cones fastened on either end. But it's actually quite effective. Now this snake trap we lay alongside the guide fence, and there's one on each arm of the guide fence. You can see snakes have an easy time getting inside, but have a hard time getting out. And the reason why is the shape of these cones, basically. The wide part of the cone, which is placed on the outside, it's flush against the ground. It's a nice wide opening. It's easy for the snake to maneuver inside. But once it's inside that tube of chicken wire, then it has to zero in on a very narrow opening, right? You have the tip of the cone on the inside, right? That is no longer flush with the ground, right? And that's really hard for snakes to do. So it's simple yet very effective, right? So here's an interesting question. I told you we take data, right? As you can see, we're measuring a lizard here. That's, again, that western fence lizard, or Scoloporus occidentalis. What does this type of data, what can it tell us? Okay, so think back, you can think about cumulatively, or you can think about specific pieces of data, right? So what does length tell us? What does weight, sexing them, right? Condition, what might that tell us about our herpetofauna at Cabrillo National Monument? What do you guys think? Go ahead and type it in the chat. How well they grow. Yeah, absolutely. That is one of the factors. You know, how are they doing? Are they growing quickly? Maybe that'll tell us something about their resources, right? Maybe that tells us something about the environment. So we have noticed in years of extreme drought, they don't tend to grow as much, right? Yeah, very well done. So in fact, here's a little shot of a paper. We did an analysis. We learned all kinds of interesting things. Health, food availability. Yeah, excellent, Karen. All of those things those are all factors that we can learn from taking this long-term data, right? And so from an analysis of our herpetofauna data from about 30 years worth of data, we can see that one of our snake species, the striped racer, and one of our common lizard species, the Belding's orange-throated whiptail, are actually showing strong evidence of declines in abundance. Right? So that means their population is decreasing, right? It's shrinking. And then several of our common lizard species, our endemic lizards to the park, are showing a significant decrease in body size. That's in both mass and length over this period of 25 years or so. So this leads us to speculate that there are some basic resource requirements that may also be in decline for these species, right? There's fewer of them in some cases, and they're getting smaller in size. But what do I mean by that, right? What do I mean by these basic resources in decline? Well, let's circle back to the idea of our national parks as a living laboratory, right? You can take that robust long-term data to really see how these ecosystems are responding to stressors over time, like for instance, climate change. And you can really compare this data with that of similar locations to check for factors like urbanization. Okay, so this is an interesting question to ask at Cabrillo because we are really an urban national park. We're on the gateway to one of the largest cities in the nation, San Diego. So the landscape and habitats of the preserved and protected Cabrillo are very different from the city that surrounds it, which you can see depicted here, right? This is all concrete and glass and development, right? Urbanization. Right, so the landscape and habitats of Cabrillo are perfect for compare and contrast with neighboring habitat. And not only that, but Cabrillo is what we call an ecological island. And what this means is that even though it's still connected to the mainland, it's not technically an island, it acts like an island because it's surrounded by water on 3 sides, by ocean on 3 sides, and dense urbanization to the north. Right? So it's really cut off from surrounding populations of the different species that live there, except for the highly mobile species like birds, right? They can kind of travel anywhere. But when you're looking at like lizards and salamanders and small rodents and that sort of thing, right? They're cut off from neighboring populations, the mainland population. And this is important to note because being isolated like this can really affect habitat quality, right? So one of the things that's positive about Cabrillo is that it is preserved and protected land. However, it's a small monument. It's only about 150 acres or so. And so fragmentation by roads, trails, and buildings, which you can kind of see from this aerial view here, can deeply affect the species that live there, right? Can deeply affect their migration and their ability to find a mate. We also have to consider low genetic diversity, right? Because they are cut off from neighboring populations. There's no gene flow going between them. And so that low genetic diversity can consequently extirpate or remove species from the park. You can have them go locally extinct. So this is something that we're keeping a really close eye on and something that we're worried about, that habitat quality for our herpetofauna, right? And here's another factor to consider and something that we're investigating actively right now as well. Is the island species versus mainland species, right, is natural selection in and of itself, right? So You're like, why do we have a picture of Darwin and Darwin's finches here? Well, remember Darwin's finches, right? How each island in the Galapagos hosts a different species or several different species from the next island. And that all came from the same ancestral species. It's called adaptive radiation, right? You have one ancestral species, they move out to different islands, and then they evolve differently because there's different selective pressures on those islands. And we're actually seeing that at Cabrillo National Monument. We're seeing that with this species right here, our one rattlesnake species, the Southern Pacific rattlesnake. We're working with researchers at San Diego State University right now, and they are taking genetic data and behavioral data from our population at the park and comparing it to mainland populations. And there's some strong evidence to suggest that there is some differences genetically and behaviorally both. So eventually, this species will, the population of Southern Pacific rattlesnake at Cabrillo will speciate from mainland populations. Will become a different species entirely. It's changing, right? The fact that we're cut off is changing our species at the park. But what else have we discovered, right? What else have we discovered? You might recall, we study a bunch of different things at the park, right? One of our methods for study is something that we call selecting an indicator species to study in an ecosystem, really zero in on that species. So another question for y'all, what is an indicator species? What might that mean? What is an indicator species? Just take a wild guess. No wrong answers here. What do you think an indicator species might mean? What does it mean to indicate something? All right. So I know, did I stump you? Okay. Okay, something that is inherent to the area that will help us get a baseline. Yes, very well done. So an indicator species is just kind of what it sounds like, right? It's a species whose health and wellness indicates the health of its ecosystem. Okay, so it's generally a species that is very sensitive to change. Okay, yeah, so how the ecology is doing, perfect. Yeah, exactly. They're sensitive to change. So if something happens to that indicator species, it could be an early indication, right, that there is some sort of problem in the ecosystem at large. An early warning sign that tells us as scientists that there's a problem that we need to investigate and then hopefully solve, right? So we do have an indicator species in our rocky intertidal zone, and that is this guy, or should I say gal, Laudia gigantea, or owl limpet. Now this unassuming little creature is a marine snail, but it's quite special for several reasons, beyond being sensitive to its environment, beyond being a really nice indicator species. First, it is what we call an ecosystem engineer, okay? So I've got a little owl limpet shell here. I'm gonna show you. You can see right here, okay? So owl limpets live their life clinging to rocks, right? And the females actually carve out a territory. And what they do is they move around their territory on the rock, mowing down algae with their specialized tongue. It's called a radula, right? But they're actually quite a powerful little snail. So not only mowing down algae as they move around, they're mowing everything else off that rock, right? We're talking barnacles, we're talking mussels, etc., right? And this may seem like it's a bad thing, but actually it makes new space for new life to come and to grow there, right? It's in nature, an intermediate disturbance, a little disturbance here and there is actually great for the rejuvenation of that ecosystem. So this ecosystem engineer is engineering the ecosystem, right? It's changing the ecosystem to make it healthier in the long run. Okay, so the second fact, the second thing that makes owl limpet so interesting is that it is something called a sequential hermaphrodite. Anyone want to take a guess at what a sequential hermaphrodite might mean? What does it mean to be a sequential hermaphrodite? You can start with hermaphrodite if you know what that term is. See, you wouldn't think this little snail is that interesting, but it actually is quite interesting. Yeah, so a hermaphrodite is an organism that is both male and female. But a sequential hermaphrodite is one that changes from one sex into the other, which I think is amazing. That'd be like me waking up tomorrow with a deep voice and a full beard and being a man, right? I mean, that's just amazing to me. So every owl limpet actually starts off as a boy. And as it grows, it reaches a certain size, and there's certain environmental cues that turn it into a girl, right? Now, this is a cool fact and all, but you might be like, why are you telling me this? Well, it actually has to do with the health of the ecosystem and how we study this organism. Okay, so keep this in mind, right? Keep this in mind, how the sequential hermaphrodism is I go into what I'm going to speak to next, okay? So people apparently really like to eat these owl limpets. I've never had one. Apparently it tastes kind of like a scallop, if you've had scallops before. But of course, at Cabrillo, they are protected. It's illegal to poach them. Harvesting is disallowed. But the species procreates through broadcast spawning. So what they do is they release their sperm and their egg into the water column, right? And then those find each other. And then you've got zygotes and larvae. And then that larva gets carried on the current, down to its new location where it starts its life, right? And since currents off the coast of California run north to south, the population of owl limpets at Cabrillo comes from owl limpets that are north of us, right? From other populations that have broadcast spawn, and they floated down, and then they've, kind of planted at Cabrillo and then gone through their life cycle, right? And they come from areas that are not protected where humans can harvest. So I think you might be putting together where I'm going with this, right? So that being said, take a look at some of this data that we've been taking over the last 30 years or so, right? So here, just to kind of set you up with a chart, we've got on the y-axis, we have the size in millimeters of owl limpets at the park, the average size. And then in the x-axis, right, the independent variable, we've got the date, right? So anyone take a stab at what the general trend is going on with our owl limpets, those Laudia giganteas? What's going on with our owl limpets at the park? Are they getting bigger or smaller on average? Yeah, thanks, Karen. You got it, right? Yeah, absolutely. They're getting smaller, right, as a whole. Now, recall they're sequentially hermaphroditic, right? The small ones are males, the large ones are females. So what does this mean? If our owl limpet population is getting smaller, what does that mean for us? What does that mean for our owl limpets at Cabrillo National Monument? Is that good for them? Is that good for the population? What do you think? I told you the questions were going to get harder as we went. Had to wear your thinking cap tonight. Yeah, no, exactly. Good guess. Yeah, very good. No, it's not good for them, right? So with most sexually reproducing species, we want what we call an even sex ratio, right? So we want about 50-50 males and females, and that's just so they can meet up, find mates, right? Make lots of babies, that sort of thing, right? So for humans, for instance, I think it's something like 49% males, 51% females on planet Earth, right? We want something similar for these owl limpets. And the problem is people are picking out the large owl limpets. They're taking the females out of the population, right? There's fewer eggs being released into the water, so fewer sperm are finding those eggs. We have fewer zygotes and fewer larvae coming down to us, and we're getting fewer and fewer owl limpets. And then what's happening is those owl limpets, right, the large ones that we did have, they age out. So the older ones are female, they die off. Then we just have a bunch of young males that are slowly growing into adulthood, right? And it's kind of this vicious cycle, right? At Cabrillo, because our sex ratio is getting skewed, the populations south of us, right, are going to have fewer babies coming to them as well, and so on and so forth, all the way down into Baja California. And you can actually see this cycle in this next figure here. Here on the left, you've got the Laudia density per meter squared, right? So how many owl limpets we've got at the park, right, in general. You can see it's separated into 3 different zones. That's just because we have 3 different zones in our tide pool. So zone 1 is the zone where most people go, most tourists go, right? And then zone 2 is a little further out, there's less human impact, and zone 3 is for researchers only, right? But if you look really closely at this, you can see, excuse me, that all 3 zones are decreasing in abundance, right? So that cycle, the older ones aging out and dying off, and there aren't very many new ones coming in, and then those are having to grow up. Like we're having fewer and fewer owl limpets as we go. So we wouldn't know this without taking these long-term data sets from inventory and monitoring programs. But what can we do about it, right? Like how does this actually help us to preserve and protect these beautiful places, right? These are rocky intertidal zone, for instance. Well, I'll give you an example. Through other monitoring in our tide pools, we were just getting an indication that perhaps humans have an impact on the environment, like tourists walking around and picking things up and rolling rocks over, et cetera. So we can actually take that information and give it to management and say, hey, we want to limit the amount of humans that go in the tide pools. And in fact, we do. So we actually have permits. There's only permits allowed for groups of 10 or more, and we only allot so many permits per low tide, for instance. So we decrease the amount of people that can actually go into our tide pools to kind of reduce human impact, right? So that's how doing science in the parks can help us to be better conservationists, can be better land managers. But surprise, I told you I would come back to this. I'm actually not in the Natural and Cultural Resource Management and Science division, even though I am a scientist. I am in the division of interpretation and education. And here is why, okay? So when you really think of it, when you distill it down, like what a scientist does into 3, you can do it into 3 tasks, right? So the first task of a scientist is to observe, right? To observe nature. And when I say nature, I mean everything from, microscopic organisms to our bodies, to other species, to ecosystems of the biosphere, to celestial bodies up in the heavens, right? Like everything in nature. So the task of a scientist to observe, and often they observe some sort of problem, right? So that's the first step in the scientific method, in fact. Then their second task is to find a solution to that problem, right? Using the rest of the scientific method, okay? So asking a question, diving into the literature to make sure that question hasn't been answered by other scientists, and if it hasn't, then you can form a hypothesis, make a prediction about the hypothesis, come up with a clever experimental design, right? Test your hypothesis, get hopefully some results, and then analyze that data, and then hopefully come to a conclusion, right? But that's just number 2, task of a scientist. And here's the hard part. What do you think number 3 is? Task number 3, what do you think that is? This is something that people always forget, and I think is equally important as the first 2 tasks. What do you think number 3 might be? I'll take a little sip of water while you think about that. And take a guess. I'll give you a hint. I'm doing it right now. Communicate and engage the public in their work. Yes, exactly. Well, perfect. Telling the story of science, right? Communicating your science. Because you can be the most clever scientist in the world and you can come up with answers to overarching problems. But does it matter if you don't tell anyone about it, right? So let me give you an example. Let's say I find a cure for cancer. Is anyone going to get cured if I don't tell anyone? No, right? I mean, you need to tell the drug companies to manufacture the life-saving drug. You need to tell the doctors to disseminate that drug, etc., right? So that's why I'm in the interpretation and education department. I would argue that communicating science is equally as important as doing science. And in fact, when you look at it through the lens of conservation, are we going to preserve and protect things that we don't know about or don't care about? No, we're not, right? So teaching people about it and getting them excited about it is equally important as doing the science itself. Educating the public about our incredible natural resources, right? So here I am educating, right? Like, for instance, our peregrine falcons, the world's fastest animal, right? or perhaps our two-spot octopus, which in many ways is more complex than human beings are, or one of the world's most endangered plant communities, the coastal sage scrub, right? Teaching people about that, showing them how incredible it is, really fosters environmental stewardship in people. It encourages them to preserve and protect these places, not only for today, but for years to come. And as you recall, with the National Park Service mission, right? Effective science communication really rallies people to spread the gospel of environmental stewardship to their children, their family, their friends, and their communities, which contributes to the mission of preserving and protecting for this and future generations in perpetuity. Okay. So here I am doing some of that. I do science and then I communicate that science to others. Oh gosh, sorry about that. There you go. Do that science and then I communicate to science to others, right? I see about 10,000 students a year. And you can see that down here in the lower left-hand picture. So you see many school groups. I do community outreach, right? Here I am, the Science Explorers Club. This is a project that I spearhead at the park where I wheel a mobile science cart around the park and engage people with fun science activities, right? We'll do outreach like I'm doing right now. I'm giving talks. We'll go to science and engineering fairs to get people pumped about science. But tonight I really want to zero in on this one that you see in the upper right-hand corner. And that is my free STEM summer camp for girls ages 9 through 16. It's called Ecologic. So this project really incorporates all the aspects of my career and my passion for conservation. It fulfills the Park Service's mission by fostering environmental stewardship and even potentially creating new scientists, right? That perhaps maybe someday will go work for the National Park Service. It teaches the next generation ways to help their environment that they can do at home and right now in their daily lives, right? So it really adds to the mission of conservation. And perhaps it even helps solve our overarching global problems like mass extinction and climate change. And it does that, interpretation and education does that, and groups like Ecologic do that really because it brings all voices to the table. Right? Sorry, I need another sip of water. My throat is very dry. There we go. Okay, so it may or may not surprise you to know that there's been a tradition of inequity in STEM fields, right? So although half of US college educated workforce are women, less than 30% of STEM jobs are women, right? So minority women in particular are underrepresented. Fewer than 1 in 10 are employed as scientists and engineers of color. So why is this happening? Well, research draws a direct connection to visibility and representation. And simply put, if girls see STEM professionals that look like them, they can imagine themselves in those roles, right? And this circles back to why is making sure all voices heard? Why is that important? How is that really contributing to the mission of conservation? Well, think of it this way, right? If all sharp minds were allowed to contribute to science, maybe we'd already have a cure for cancer or a solution for climate change, right? So this is why organizations like the American Association for the Advancement of Science and Lyda Hill Philanthropies are spending a considerable amount of time and effort to decrease diversity in STEM fields by creating initiatives such as If Then. If Then is a slogan that refers to if we support a woman in STEM, then she can change the world. And as was mentioned earlier, I happen to be an If Then ambassador. I have the honor of being one of 125 women in very diverse STEM fields across the nation to help kind of bring about that culture shift, to be able to bring all those voices to the table, to change the narrative about women in STEM. And I'm telling you all this because being able to do this has given me the opportunity to encourage girls to pursue STEM and to foster that environmental stewardship on a much larger and perhaps even more meaningful scale than ever before. Like, who knows, maybe one of these girls will grow up to be a lecturer in the Sharp Minds series, right? maybe one of these girls is actually even one of you tuning in right now. Well, thank you very much. It looks like I'm just about out of time. Thanks for coming to learn how the Park Service preserves and protects. Definitely have some time for questions here. Anything's on the table, you can ask me about being a conservation biologist, being a science educator, being an if-then ambassador, how we monitor in the parks. Whatever questions you have for me, I am here to field.
Wonderful. Thank you so much, Samantha. Also, thank you for that nice segue and reminding me that I should put the, is that coming from my background?
That's probably my cat. Sorry.
Oh, okay. No, that's okay.
He's very vociferous.
My husband doing.
He likes to talk.
Why not? All voices should be heard. So because Samantha is part of the If-Then initiative and she's an If-Then ambassador, we are having a very specific survey for our participants tonight, which I would really love for you to take because the If-Then initiative and the Fleet Science Center would like to learn a little bit more about how If-Then is perceived, how effective it is, how it affects people in presentations. And we also want to know if you enjoyed this event and the presentation in general. So if you could take a moment to take that survey at the end of the night. I just put it in the chat. So if you click on it, then it opens up. It should open up, hopefully, if you can't click on it. If not, you have to copy it out and put it in your browser so you don't forget it to fill out later. Because now is question time, so we don't want to sidetrack you with a survey when we can ask fantastic questions of Samantha. So we have one question already, and that is, can you collect limpets from the north and take them to Cabrillo? So, you know, not that, yeah, can you?
Yeah, that is a absolutely great question. So the answer is yes, we could do that. And that is not something that's off the table. But before you want to go ahead and do something like that, some sort of restoration work like that, you definitely need to study all of the different factors of like why our populations might be declining. Now, we do think it's because of people harvesting to the north, right? But there are other factors as well, such as climate change, like ocean acidification, right? So right now we're just kind of in the gathering information phase of things. But once we've kind of like really discerned what's going on with the ecosystem, then we can really come along and come up with some solutions to the problem, right? And that's one of the things that I think is probably going to be bandied about, right? Like restoring our population at Cabrillo and bringing in owl limpets and releasing them at Cabrillo, right? Great question. Any other questions for me?
Yeah, I have one actually, if you don't mind. So,
yeah, of course.
So this is one of the, there was the word which I've already forgotten, but it's kind of the baseline species. Do you have others? I mean, because this species is dependent on what's coming down to Cabrillo from the north. But what about the other species that you monitor if you see the numbers going down? And you mentioned that some of the numbers for the lizards and the snakes were going down. Mm-hmm. What is your thinking around that, and what kind of solutions do you have in mind? Where do you think the problem sits?
Yeah, that's a great question. So we do, bringing it back to our Rocky and our tidal zone, our owl limpet is not the only thing we monitor there. It's just the one thing I talked about tonight. So we do actually do like full biodiversity surveys with transect lines and with quadrats, right? These are tools that we use in our trade that we really look at all the different species that live there at the park. And it's impossible to count everything that lives there. So you go back, you have specific plots that are scattered throughout, and you go back to the exact same locations year after year and take that data so you can actually compare how it's changing from year after year. So yeah, so owl limpets are great because they're sensitive, they're that indicator species that's sensitive to their environments. That kind of helps us get some early cues, maybe some early warning signs. But we are keeping an eye on pretty much everything in the park as well. And you asked a really good question, like, what do we do about our lizards and our snakes? And really the answer is we don't know quite yet what it is we're going to be doing, because we are cut off, right? Cabrillo is not going to gain any more land, for instance, right? It's completely urbanized to the north of us. So our populations are probably just going to have to run the course, their genetic course out there. And what we can do is make decisions like where we put trails, right? To avoid more habitat fragmentation. We can restore native habitat by planting native plants, right? If there's a drought that wipes them out. We can do things like getting rid of our invasive species, which are, really detrimental. So for instance, we used to have a coast horn lizard at the park, which we think is extirpated, right? We haven't caught one in many, many years at this point. And we think that's due to the invasive Argentine ant actually coming in and outcompeting the ants that horn lizard really likes to eat, right? So we can do sort of management like that, but the fact of the matter is the population is going to remain isolated. And so we'll just have to track what happens as it speciates from mainland populations, right? And see who's able to adapt and who isn't. All we can do is mitigate, but not stop, if that makes sense.
Yes, it does.
Good.
Samantha, I'm going to stop sharing your screen right now.
Oh, yeah.
So we have a better view of you.
Great call. There we go.
Perfect. There we go. Yeah, it's just easier for attendants to see you when you ask the questions. So I have always plenty of questions, and I'll continue asking them until I see some more in the Q&A or the chat, but please go ahead and ask us your questions. You mentioned the trails. Of course, we want people to know about Cabrillo Monument. We want people to fall in love with it and care about it. How do you see having a lot of visitors impacting the ecosystem, and how do you try to balance that? You mentioned that you limit for the tide pools, but the general trails, are there any things that you try to make sure that we're not harming more than we enjoy?
Yeah, absolutely. Well, we always ask that there's no picking or taking of any kind, right? So do not take any rocks from the tide pools or shells. Do not pick any flowers, that sort of thing. Leave things as you find it. The trails that are already cut, right? Those have already fragmented the landscape. But if you stay on those trails and you don't pick things, right? You're not trampling plants, you're not damaging our natural resources. And that's really the best way that you can preserve and protect these places. And it is definitely a balancing act in the Park Service, right? Because we want this to be a fairly intact ecosystem. We want it to be its beautiful living laboratory. We want a place for wildlife to be able to flourish, right? But these are also people's public lands, and we also want people to come and enjoy them and care about them so that they will continue to protect them over the years. And so really trying to balance that, right? So anything that we do at the park, we have to follow federal guidelines, right? So there's policies that we have to do really robust surveys to see how decisions like building a new trail or building roads will impact the environment, right? And take that into consideration. And many of these sorts of decisions are open for public comment too. So we actually open it up to the public to be like, hey, what do you think about us cutting a new trail to the tide pools, for instance, which is something that is being considered actively at Cabrillo. And, we listen to that, right? So if the public is overwhelmingly like, no, we don't want this because it fragments habitat, or if they're like, yes, we definitely want it, right? We'll take that into consideration as well. So, again, it's that fine balancing act, trying to meet all the needs of the stakeholders involved, right? Which includes wildlife and humans.
Yes, but it's nice that the public has a say also. I think that's great that we can be stewards for that area as well. We have another question. Do you know how many Argentine ant super colonies are at Cabrillo?
That is a fantastic question, and I love it. So Karen, you really know your scientific stuff, and it's been a pleasure having you participate tonight. So we don't know how many super colonies there are. We do know they're there because we've done a couple of pretty robust ant surveys with the David Holway lab from UCSD, which is an entomology lab. And we actually harnessed citizen scientists. We did kids from high tech came out and collected ants for the Holway lab with us. And so we discovered that we actually have 11 species of native ants, which is great news. But we did also catch some Argentine ants, so we know they're there. And fun fact, which may be unsurprising to some, is where we found these Argentine ants tend to be where people were at, right? So around our main parking area, around our visitor center, because again, people are vectors for invasive species a lot of times. So those ants rode in people's cars or backpacks, right? And have gotten into the park. But the good news is they don't seem to be as abundant as they could be, certainly as they are in much of San Diego. And those other species of ants are still there and thriving. So hopefully the super colony has not taken over yet, right? Yeah, that's a great question. We're basically San Diego is basically on top of one huge Argentine ant super colony. It goes for miles. It's pretty crazy. These ants are amazing. Yeah, they're amazing.
Maybe there's something special about Cabrillo that they're not taking foot there. Maybe there's something they don't like about Cabrillo. That they're not
Could be, yeah. I mean, again, it's far out on the peninsula, so it's also harder for them to reach, right, over time. So, yeah, great question. And I often get asked, well, what do you do? Like, how do you get rid of them? And this is the problem, right? I mean, you can't. If you use insecticide, it's not species specific, right? So if we were to use some sort of insecticide that killed ants, it would kill off our native ants, and they are, of course, very important to their ecosystem, right? They're an important food source. They're an important decomposer. They till the soil, for instance. Like there's many things that they do. They pollinate, actually. I don't know if people do that. Not every species of ant does, but many of them do. So we want to. How do you get rid of the invasive one without getting rid of the native ones, right? And that's kind of an age-old question. Hopefully we'll find an answer to that someday. Not there yet, but someday.
Are there any specific projects like citizen science projects that one can get involved with Cabrillo if we wanted to help?
Thank you for asking that question. Because
and you didn't take it to that. Just
I didn't. No, in fact, there is one going on right now. Every year, I tend to do a big 24-hour bio blitz at Cabrillo. And we do that during the City Nature Challenge at the end of April. But of course, this year with the shutdown and COVID and, didn't want large gatherings, that sort of thing for health and safety. We did a backyard bio blitz where we just encourage people to kind of go out into their nearby nature. But right now, what we're doing is the Parks for Pollinators bio blitz for the entire month of September. So you come out to Cabrillo and you take pictures of pollinators, and then you put them on our iNaturalist Parks for Pollinators page, and then we are taking people's observations and posting them on our social media. So maybe your observation will be famous on the Cabrillo social media if you come out and do the Parks for Pollinators events. But definitely keep your eyes peeled. If COVID allows and it's responsible and safe, I'll definitely be doing another 24-hour bio blitz come April this year for City Nature Challenge.
Awesome. Great. Thank you so much. That also brings us right to 8 o'clock. And I think that is a nice note to end it on, that we can all help and do our part in research and conservation.
Absolutely.
Samantha, thank you so much for your time and your talk today. I really enjoyed it. Thank you so much. I really appreciate it.
Thank you for having me.
Of course. And to our audience, thank you so much for coming out tonight, for interacting with Samantha, for answering her hard questions, for making this a very personal talk, which I appreciate. Stay connected through our website. I'm going to put it in the chat one more time, just in case. It's rhfleet.org. Pretty easy. We're also on social media. We're on Instagram. We're on Facebook. We also post our events on Facebook. So if you want to find a way to stay up to date with all of the events coming up, those are good ways to do that. And with that, don't forget to take the survey, please. Thank you.
Thank you, everyone.
Thank you so much. Have a great night. Samantha, if you could stay on for 1 second, that would be wonderful.
Will do.
Thank you so much. I'm going to